Hello, I'm Joe Barry. I'm here today with a panel to talk about a new area of science called a reasonably new area of science called physiological ecology that was developed in the 80s and 90s, started in the 60s. The panel that's here today and will discuss with me is Christopher Field. He's director of the Department of Global Ecology at the Carnegie Institution at Stanford. Olle Björkman, a retired member of the Department of Plant Biology at the Carnegie Institution, and the former head of the Physiological Ecology group at Carnegie. Barry Osmond, retired director of the Research School of Biological Sciences at the Australian National University, and was president of the Biosphere 2 venture when Columbia University ran it in Arizona. And Harold Mooney, professor of biological sciences at Stanford, and a recent winner of the Volvo Prize for Environmental Sciences. So the topic of the day is physiological ecology. And I'd like to start with just a little bit of background on what physiological ecology is. It's a branch of plant physiology that seeks to describe and understand the physiological basis for the performance of plants in the context of their native environments. It tends to view plants as an integrated system as opposed to being focused on any one particular thing like respiration, photosynthesis, water relations. And while it's focused on plants in native environments, it's also very relevant to understanding agricultural production. And it's provided a basis for scaling up from studies at the organismal and biochemical level to larger scales, particularly to the global scale. And it also has application now in trying to relate the advances in genomics to the kinds of impacts that genomic differences have. The science of physiological ecology arose maybe 25 to 30 years ago in a context where specialization was viewed as being kind of the good thing to do in science. And most of the scientific ideas and problems that were worked on at the time were kind of self-generated within the disciplines. However, at the same time, there were a number of things that kind of cut across disciplines, things like the phytogeography, distribution of plants, functional constraints on the distributions of plants, and particularly evolutionary biology, trying to understand how these differences in plants were related to their evolutionary background and their genetics. And ultimately, these broader interests kind of broke loose and inspired people to jump out of their boundaries. And the group that's assembled here were really instrumental in causing this breakthrough. So they sort of tipped the balance to a systems approach. I thought I'd just give one example, as shown in this first slide, of how an example of how physiological ecology, a kind of problem that it has addressed. In the time frame of about 1965, Olle Björkman discovered that oxygen inhibited the quantum yield of photosynthesis. That is, that more quanta were required to fix a CO2 in normal oxygen than in low oxygen. This effect became somewhat more complicated in about a year later, when it was discovered that there were plants that lacked this inhibition. They were called C3 plants, C4 plants. The C3 plants were inhibited by oxygen. The C4 plants were not. These plots, the slope of these lines in the upper left-hand corner here, indicates the number of quanta required to fix CO2. C3 species, this quantum requirement is sensitive to oxygen and to CO2 concentration. And with C4 plants, this is independent of oxygen, but it is dependent upon temperature. These responses can now be understood by the finding that the enzyme which fixes CO2 is also fixes oxygen, and this propensity to fix oxygen causes this inhibition of photosynthesis. And C4 plants lack this effect because the CO2 fixing enzyme ribisco was protected in C4 plants by a CO2 concentrating mechanism. Now, this is the discovery of this phenomenon of the oxygenase of ribisco is really a biochemical discovery, but in fact it was based on and inspired by studies of the oxygen effect on photosynthesis, so that it came from physiology, not from biochemistry. This discovery had some important implications for how C4 plants differed in their adaptation to environments than C3 plants. C4 plants, based on these studies, might be expected to be more successful in warm environments where temperatures are higher than, say, 25 degrees C during growth, and C3 plants more successful in cooler environments. These differences are captured actually in mechanistic models based on the carboxylating enzyme, and maps of the distribution of C4 plants can be constructed from the knowledge of where they might be expected to be most successful. The models are incorporated into global Earth system models, so that now we know where C4 photosynthesis occurs, where C3 photosynthesis occurs, and what the quantities are that are associated with these. It turns out that it's probably someplace between 20 and 30% of the total photosynthesis of the world is by C4 plants. We also are able to check this by other findings that have come out of physiological ecology, which use or utilize the isotopic preference of C4 plants for higher proportion of carbon-13 in their biomass that they synthesize than C3 plants. So the idea here of this explanation is that it illustrates a way that the systems approach, which has come out of physiological ecology, has been useful in making a link between scientific investigations on sort of small scales and across disciplines, and bringing this to larger scales all the way up to the planetary scale. And I don't think this is something that would have been generated by any one of the disciplines, photosynthesis, traditional plant physiology, all by itself. So that's I think, an accomplishment of physiological ecology. And there are many ways in which this kind of systems thinking about biology has been a part of the recent development of studies of the global physiology in agricultural and global systems. Now, I thought it'd be good to start our discussion here about where this is going. I think one of the things that's really important here is that all the folks here have made really important contributions in the area of physiological plant ecology, almost all gone on to larger scale systems and making contributions to understanding the global system. And I think this reflects the urgent challenges that we now face at the global scale with food security, population, climate change. And I think that the successes that we've had working through the science of system science of physiological ecology upwards towards global systems makes me optimistic that we can actually contribute importantly to the science of understanding the issues that we have to meet at the global scale. So with that introduction, I thought maybe we could say a little bit about the global system. Chris?
Well, I'd love to hear a little bit more about the intellectual environment during the era that C4 photosynthesis was discovered. Was there a sense that everybody was working together on a shared sense of issues? Was there a sense of competition? When did people really know that a big breakthrough was on the horizon?
I've always been impressed with the area of physiological ecology, which has not, in my view, has not been so competitive, but has been cooperative. And it's remarkably different than many areas of science. And I don't know why that came to be, but it's true. And I think that's what's helped our progress in many ways. That's what you asked, is that right?
Right. Yeah, that's my sense as well. I think that even though people knew that there were other groups who were working on the same problem, that are able to approach it in a way that said, maybe we can achieve more together than if we were doing it separately.
It's been remarkable.
I think in part that's because they were at different levels or had different expertise. And one of the things that we all experienced in this is that you were going out of your depth in one area to try and solve things that were like the oxygen inhibition of photosynthesis required enzymology as well as gas exchange.
So it is an integrated science. Right?
So we're interdependent.
Right, interdependent.
And I think there also was the fact that there were no big prizes out there.
Right.
Somebody thought they were going to win if they could just beat the other guys.
Well, I think if we go back to what Joe mentioned about C4 photosynthesis and the area of photorespiration, oxygen inhibition, and all of that, it was an unusual time. We got a lot of collaboration going very quickly, very open communication. No one was secretive about his or her results, and there was a very free exchange among people, but also from different disciplines. And I would take the real breakthrough was in 1970 when Barry Osmond, Rel Slatcher, and others organized a conference on photosynthesis and photorespiration in Canberra and invited people from all over the world in with different areas of expertise and so forth. And we had 10 or more days of intensive discussions, often extremely open and collaborative. And that also very much strengthened the whole collaboration later on and the development of different groups.
Yeah, I think this is so, Olle, that meeting is widely regarded as being a sort of a melting pot, and then there was an explosion in the pot, and it worked extremely well. My guess is that an element in making that work so well was that, first of all, there was a lot of biochemistry going on around the world, particularly in the US, with respect to things like the anatomical description of photorespiratory interactions and all of those things. But at the same time, the two of the drivers were here in Carnegie and in Canberra at the Research School of Biological Sciences. You know, I was just a junior non-tenured faculty member, but in both situations we could do what we wanted to do without having to fight for peer endorsement and funding sources outside of the institution. I think this mattered a lot because there were some referees of papers in those early days that said this paper is unpublishable. It assumes that there is a process called C4 photosynthesis. And you know, there was a hell of a lot of antagonism from certain sectors of plant biology, but the freedom that we had and the excitement of pulling things together, I think, were major factors.
How long was the period when it was clear that there was something different that was happening that couldn't be explained, and where the framework of C4 hadn't really emerged, but it was simply a series of not disorganized but disparate observations that didn't fit together?
I
Joe,
don't think it lasted very long at all.
yeah.
Well, it was in 1966, the first really credible publication that showed that CO2 fixed by into C4 acids moved on through the photosynthetic sequence. When that was published, it was that was 4 years to when essentially almost all the I's and T's were dotted and crossed at the meeting in Canberra.
Yeah.
So it happened really fast. And there were a lot of other things put together related to the anatomical association of C4 photosynthesis with the bundle sheath anatomy, Krantz anatomy, and the association with the oxygen inhibition that also kind of fit into place. And then the really key piece that solved it all was the discovery by Bose and Ogren that CO2 fixation by ribisco was inhibited, and then that inhibited by oxygen, and then in addition to that, there was the finding that it produced an alternate product.
Well, you were also part of that discovery. I would like to go back for a moment to Barry's about that we had a certain level of funding, so we could act very quickly. Now, this was particularly true at the Carnegie Institution, Department of Plant Biology under Stacy French. He was very frugal, and our budget was very limited. However, we had total freedom to do whatever we wanted. We could act very quickly, and as soon as you got an idea, you could go into the lab and start making the preparation and construct the equipment that you needed for the work. And there was no one hindering you in any way other than possibly lack of money, but that was it. And then a wonderful atmosphere to work in. And I think that is not longer present in most places.
There are very, very few refugia of that sort. I think the Max Planck Institutes, from the little bit I can observe of them in Germany, they still manage to perform in this way, but nearly all of the institutions that had the freedom that we had in those days have disappeared, and the Australian National University has been deconstructed in that context completely. Yes.
Do you think that kind of financial and administrative freedom is critical for the assembly of an interdisciplinary integrated science?
Well, I personally think so, and there's been a lot written recently of the problems of this sort of bibliometric, factor-driven research decision-making by people who are not in research, but administrators. And some of the stuff I've seen, in fact, the bottom line that emerges from that is that we need to return to block funding. And the block funding would be of the sort that might have been free and accessible in, say, the Department of Energy in the early days. In independently funded institutions like the Wellcome Trust in medical research in Britain, there's a growing sense that unless we move this way, we're just going to be ground down by the administrative management of resources and management of ideas. Yeah.
You were at that symposium a couple days ago where we saw a really bright light sort of starting to blink in this regard. This is the Mellon Foundation, which is for 20, 30 years now has invested in young people with bright ideas. And that was the only requirement. You send us your bright idea and we'll fund you. But now this Bill Robertson, who ran that program, is retiring, and I think they're going to be changing to a different direction. And then there was the other foundation, which was the Keck Foundation. It was the same model, and very important.
Yeah.
You know, we've talked this morning about two really important formative themes in physiological ecology. One was the theme of understanding ecotic differentiation, which I think came mainly from a field-based and a genetic kind of a framing of the issue. And the other was the discovery and elaboration of C4 photosynthesis, which really came from the biochemistry-chemistry perspective. And one of the things I think is really exciting about the way the field emerged is that those two different lines of thinking really fit together, so that the ecological and genetics really fit together very well with the biochemistry and chemistry, and really laid the foundations for what physiological ecology became.
One of the really important additional aspects of this is the ability to get out in the field and actually make measurements of what plants are doing in the field, and to sort of make the connection between these laboratory scales and the discover and how the plants actually perform in nature. And that's an area that you really led the way, Hal, in developing the mobile lab and the Death Valley experiments that were done.
Yeah, well, I think that one of the other elements of the development of physiological ecology was this the emerging capacity to make measurements, critical measurements in the field of physiology and microenvironment, and all these things just sort of came together in the 60s, and that was a great advance. And then in addition, the advancement and development of controlled growth facilities, where you could do essentially the Clausen, Keck, and Heise experiment, but in a controlled environment. And then you could actually do all three, as Olle led, and Death Valley experiments, that the controlled environment plus the field experiments, the common garden, plus the physiological measurements in the field. And that just brought an enormous wealth of information you could integrate and come up with some answers about why things were doing what they were doing and where they were doing it. But the physiological, I come from the ecology side, and I was extremely fortunate to fall in with this group here, you, Joe, and Olle initially, who brought their enormous expertise and leadership in biochemistry and physiology, and I brought more of the ecological side. The ecological, I guess, driver was a little different. We're more interested in the whole package, how the whole package fits together, and the trade-offs that are made within a plant for adapting to a given environment. It wasn't one single process, but it's a bundle of processes and the trade-offs between them. And I think that what was happening in the 60s, probably, the energy balance development, the water balance development, and then the carbon balance, it all sort of fit together, where you could then understand what's controlling the distribution of plants and what the trade-offs are made, and who the winners and losers were, and why that was so. So that's sort of answers your question, I think, a little bit.
Yeah. Now, do you feel like you and your colleagues in that era were deliberately going out and trying to build these multidisciplinary teams, or was it more that If you had an exciting question, you would just look for the person who had the expertise and kind of build things on a step-by-step basis.
Well, physiological ecology is an integrative science. And you can't, biochemists can't do it alone, physiologists can't do it alone, ecologists can't do it alone. You have to do integration to do anything really high quality. And I think that those people who've been successful have really reached out to try to build those teams. Of course, I've always been interested in integrative science because it's so exciting that you can do so much more as a team than you can do as an individual. And it just builds the strength of science of working together. Individuals can make breakthroughs and very important, but I think integration, anyone can join into that and contribute and make things advance our knowledge.
I'd like to comment a little bit on that. The success then in the mid-60s and early 70s, when we all worked together, really, it was not top-down management at all. It just people who had similar interest and learned about what the other guys were doing, and we all became instant colleagues and friends and carried out the work. There was absolutely no administrative structure involved of any sort. And that doesn't mean that I don't think such a thing is needed now with the complexity of global systems, and where you need a lot more people and very many more disciplines involved in the work. But if I just say how it worked in our time, and that was just grassroots stuff, and it was not managed in any way.
We're losing that, and this is what's bothering me a lot, is we're seeing more top-down from the National Science Foundation, we're seeing more top-down from private foundations.
Yes.
It's we're losing this the spirit that made the kinds of work that we did successful, and I'm unhappy about that.
Yeah.
How important was the field context in terms of developing those relationships? A lot of times the research was done out in remote locations, and there really wasn't anybody to talk to around the campfire at night except for your scientific colleagues. And I think that's a very different setting than one where you're working in the lab during the day and you go home and see your family. Or
Good point.
Good point.
I don't know.
Well,
That's a great story. So
yes, indeed.
Around the campfire.
I guess the work that we did in Death Valley in summer wasn't really inducive to any campfires, because it was very hard. But it was one time where you had really to endure the climate and to be together with the plants to find out what they were doing, so it became a necessity. And I think we never had as much fun as we did together when we were down in Death Valley carrying out these experiments with a mobile laboratory, and then transplant stations there and elsewhere, and then coming back to the lab and continuing aspects of the work. It was total enjoyment, and for me, most of the work I've been doing as science was just fun. It was really a lot of fun. And the same thing, I think, in Canberra, when you started to move out into the field also, and we went on field trips in the quite not to the same extent, but they were very important.
Adding to that, what Olle was just saying is that one of the characteristics of I think the early physiological ecology was to work in severe environments.
Yeah.
Because the theory was the biotic interactions were lessened, and you could look at the direct interaction of the physical environment with the biology. So that means all the places you worked were pretty severe, and so you didn't sit around the campfire. It was more survival. We were, just as one example, I thought we had it made finally. We're going to get a nice environment, So we started working for the coldest environment you could find for where plants grow in the summertime, which was Bodega Bay, a beautiful site in California, right on the beach. And it was it was worse than Death Valley.
I remember you sitting there.
Joe and I have been trying to locate that maxim that from a long way back, that ecology is physiology under the worst possible conditions. Can you help us later track that to?
What was the?
Ecology is under the worst possible conditions.
Oh, really? No.
You don't know this one.
No.
Oh, okay. Well,
come from a physiologist. Another thing that was quite amazing about those early experiments is that you had to build almost all the equipment, and really a lot of the time was constructing and imagining and troubleshooting the stuff that you used for the experiments.
Well, now you don't have to do that so much. Some people, of course, still do it, but it was a necessity then. It was more fun, though, to build and tinker with equipment than just sending in endless proposals to various organizations to get funding to buy them. So,
one of the surprising things that I found about the field experiments was the assumption that I think was rather common at the time, that plants in the lab were going to be really much more productive and in much better shape than plants in the field, and that you would just see stunted, barely surviving plants, and the trips to Death Valley were remarkable in the sense that they produced the highest rate of C3 photosynthesis at the time, the highest temperature optimum and highest rate of photosynthesis of C4 plant at the time. Plants in the field turned out to be really capitalizing on their capabilities in the environment, and we really had to learn to grow plants well in the field, in the lab, to duplicate what we were seeing in the field.
and that took a bit of doing.
And you were remarkably successful at doing that, I might add.
Yeah, well, the big lesson from all of this fieldwork, of course, is that plants are really much more clever than the people that are studying them, and you just never know when you're going to stumble across something that really, changes the way you think about things, and that's happened repeatedly in ecophysiology, and it's had a huge impact on photosynthetic metabolism in general terms. Both the light and the dark reactions of photosynthesis. Olle sort of saw that need to pull it together right from the beginning, and then I guess the more complex system stuff that you guys have done with nutrition and ecosystem interactions that always were suspected, but it's these studies that provide the experimental evidence that it is so. Yeah.
I wonder if you want to say a little bit more about that. We tend to think about physiology as driven primarily from these well-understood model systems where if it doesn't happen in spinach, it's not worth knowing about, kind of. And it really does seem like a lot of the breakthroughs in plant biology broadly over the last several decades have really come from observations that started out as field-based or as ecology-based, where some plant was doing something strangely and people said, boy, we really got to understand this.
Well, it was pretty much the way the succulent metabolism followed on from the C4 studies, that we knew these plants were different, but we didn't have a great deal of understanding of how, for example, a cactus from Louisiana could occupy in the course of 10 or 20 years an area of eastern Australia that was equal to that of the British Isles. I mean, these sorts of invasive events focused our attention on what was it about a cactus from Louisiana that made it possible to do it there and not there, and all of these sorts of things. These guys worked with. I don't know when it started, but most of plant biology today is Arabidopsology. It's associated with this tiny little weed. Joe, in the 80s, went out in the backyard in Canberra and found a relative of Arabidopsis that is just as tough as all get out and likes to grow between the slabs of poured concrete around greenhouses and so on. And this plant, I think, still holds the record for rates of carbon assimilation in C3 plants. There's this Hirschfeldia. Yeah. Frankly, I've said it to you before, I think, Chris, with time on its hands, natural selection has had a lot of fun and uncovered things that we just aren't able to reconstruct using targeted mutations in Arabidopsis. So at the same time, we can use these targeted mutations to understand mechanisms. And Olle has done a great deal of work in recent times, and we'll talk with Krish Nayogi about this on another occasion. Yeah.
Can I come back to one thing you just said, Barry? I hadn't realized that the work on elucidating the pathway of CAM photosynthesis was really driven by a desire to understand this extreme ecological invasion that had occurred in Australia.
No, I've put a gloss on it, a spin on it that is probably inexact. The first thing was, hey, this looks as though elements of C4 photosynthesis are working differently in CAM. And I mean, the succulent metabolism was all about peculiar respiration. That was the focus until after C4 photosynthesis. And then, did this contribute to? And there's no question that the empty niche in Eastern Australia was one that could be exploited with an organism with these properties. It did depend heavily on the absence of natural predators as well. Yeah.
One thing that came out of the physiological ecology area of sort of theoretical importance that is the concept of optimizing resource utilization. And I wonder if you could comment a bit about how that's been of importance in scaling up from the small-scale studies that we initiated to trying to simulate photosynthesis and respiration of large-scale systems.
Yeah, my impression is that from the very earliest days, Hal and Barry and Olle and Joe were all thinking about plants as sort of crafty green strategists where they've got some limited pool of resources and natural selection has shaped them in order to use those resources to do as much as they could do. And if you understood what the rules were that regulated how much plant growth or how much range you get out of a pool of resources, you could predict how those plants were going to do. And I think that a lot of this background stuff we've been talking about with the elucidating the photosynthesis pathways and the role of nutrients and the role of water balance has really laid the foundation so that we can essentially make that conversion from the pool of resources to the plant performance. And, beginning, I don't know, in the late 1980s, Hal and others said, if we really want to understand these global change phenomena, we need to have a systematic way to understand what's happening in the biosphere. And, I think as early as around 1980, Hal was saying maybe these resource use efficiencies, the transfer equation from how much growth you get out of a pool of resources is a foundation for doing that. That was really inspirational to me, and a lot of the scaling work that I tried to do was based explicitly on saying, okay, given that we learned a lot from the work that you guys did in terms of how many resources you need to build a plant, let's use that exact equation to move toward continental scale or global scale vegetation processes. And as skeptical as I was about whether that would work in the very first, it actually has worked extremely well. And when we look now at the modeling approaches we have for things like global total plant growth, they're still based in the exact details that we took out of those early physiological studies.
Yeah, I want to emphasize that. I mean, that was an enormous breakthrough because it brought, we're all trying to construct an Earth system science, how the Earth as a system operates, and the geophysical people operated at very large scales, the global circulation models, kilometers, and we couldn't bring the biology, the kind of individual leaf level, to that scale. And I think Chris's work and others has managed that breakthrough where now biology and the biophysics are really equal in the Earth system modeling. It took a while for them to figure out they had stomates, as you know, you remember that. And then they came to roots are important too in terms of where the water comes from. And so those simple processes are now built into the global models, and the work that Chris has done on allocation, and particularly his discovery of the relationship between nitrogen content of leaves and their photosynthetic capacity, which is now built into virtually all of these large-scale models. It's really remarkable progress that's been made.
To what extent has the terrestrial biosphere got into the general circulation model?
It started in about 1990. That it was recognized by the atmospheric scientists that it was important to be able to simulate the stomatal conductance, essentially the surface conductance, the resistance to evaporation of water. And we made the argument that you couldn't do that in isolation. It was totally intertwined with photosynthesis. So that, and since we were able to put those into equations, the atmospheric modelers were happy to put it in as long as it didn't use too much computer time. So it was sort of getting a foot in the door, or a Trojan horse. For something they wanted to do, we got something we wanted to do, putting photosynthesis initially into the global models, and then based largely on Chris's work on respiration and control of respiration and decomposition by pool sizes, we've been able to put the complementary part of the carbon cycle into these large-scale models. So the models that we now run have reasonable representations of ecosystems. There's still a lot to do, but it's been remarkable progress, I think. Well, I think that this is a reasonable time to make a break here, since Chris has to go on to travel to give a lecture tomorrow. And thank you very much for your help in this conversation and the brilliant insights that you've brought. We'll take a short break and regroup after wishing you well.
Well, thank you very much. I really enjoyed the chance to sort of have a little stroll down memory lane and to remember how a lot of these early things really are still making a difference in the work we do today.
Still making a difference, right?
Welcome back. We're back to talk about physiological ecology, and one of the key elements in the start of this was really the first real review of the topic was published in 1957 in the annual reviews. It was a chapter by Dwight Billings, and Hal, you were a student of Dwight's at that time.
I was Dwight's second student, actually. Larry Bliss was his first student. How I got to work with Dwight Billings was as a freshman in a junior college. I read an ecology textbook, a botany textbook, and read the ecology chapter and said, gee, that sounds really interesting, this ecology stuff. And at the time, the instructor was going to be the director of the Rocky Mountain Biological Lab. And he asked if I wanted to go for the summer to take a course there. And I took my first course in plant ecology. And on the way out, incidentally, we drove out from California and going through the Intermountain Range, and you saw the zonation of the vegetation. And that to me was so interesting. What controls the zonation of vegetation sort of became a passion with me. And so then I took this course, and then as a result of the course, the instructor, Harriet Barkley, recommended me to go to as a research assistant the following summer at Niwot Ridge in a project run by John Marr. This was one of the first this was in the 50s, early 50s, one of the first government-sponsored research programs in ecology in the mountains, and it was sponsored by the Quartermaster Corps. And John was a very good ecologist, and I learned a lot. And we were doing mainly setting up weather stations up and down the mountainside to understand the climate of the area. Well, I became very, I loved it there. And John, at the end of the summer, Dwight Billings came through to visit, and John said, for graduate school, you ought to go work with Dwight, and because he's developing this new field, and there were only 2 areas in the United States at that time where they had a graduate program in plant ecology. Mm-hmm. One was Duke and one was Wisconsin. Mm-hmm. So John I respected highly, and I said, I'm going to go work with Dwight, and so I applied, and that's where I ended up. Now Dwight was he was just moving from the University of Nevada to Duke University. And while at the University of Nevada, he had done some really interesting experimental work, trying to bring physiology to the field, why certain kinds of plants could grow in these altered andesites. And he sort of developed these ideas of doing experiments in the field and in the lab, and was at that time, he wrote the first review called Plant Physiological Ecology. So I went with the right place. But his philosophy for his students was go out to the West and study the physiology and ecology of all these various wonderful systems there. So each student would go to some different place. There wasn't a team, there was you, and you had to learn on the fly. And I got very fortunate and worked on a project which led me to eventually to Carnegie, was looking at the distribution of an Arctic alpine plant through its full range, going from the southern Rockies all the way up to Alaska, and asking whether there was ecotypic differentiation of those populations. So in the Billings tradition, we made measurements in the field, brought the seeds back, and we had to construct our own growth chambers out of freezers because we didn't have, the grants at that time were very small. And so that's what got me interested and involved in physiological ecology, and we're using for the first time in the field infrared gas analyzers in the United States anyway, and we were using all the techniques which then became very central to the field. But we were doing it, I think, in a fairly crude, primitive way, for sure. And then I went to from there to when I graduated, I went to UCLA and started working on mountain zonation, one of my first loves. And we did a lot of work in the field, the same techniques and approaches. But then I had the opportunity to go to Stanford, and had met Olle in UCLA when he went through in his way from Sweden to Carnegie. And he convinced me to going to Stanford would be a good thing to do. And that was the best move I ever made. I can't tell you how fortunate that was for me, because I got to work with Joe and Olle and use the sophisticated approaches that they were using, which was far removed from what the ecologists were doing at that time. And so I think that working together, so I brought more the ecological dimension, something they didn't have as much as my training gave, but they gave me so much more in terms of the understanding and the techniques and bringing the answers to the questions that I could put in a different context to answer different sorts of things. So that was my road to Stanford Carnegie, and through Billings, who was a pioneer. And he trained so many students who went out to the West and tried to understand what was going on. But they didn't, in the end, have the sophisticated techniques that I was able to gain by joining the Carnegie group.
You went on, you sort of used, it seems to me, physiological ecology as a springboard in other areas. You went on to do convergent evolution and invasive species, and
the convergent evolution was just an extension of physiological ecology. Is that that the idea was in a given environment, a given climate, there's certain adaptations which would come to the fore, and that so if you had the idea was if you had the same kind of climate, we already knew that plants looked alike. They were evolutionarily distinct. They looked alike, but they had different evolutionary history. Did they operate alike? Was the same? It was sort of like the Clausen, Keck, and Heisey original transects, where they grew plants and found different responses, but they didn't know the mechanism that led to that. Well, the convergent evolution was the same thing. They knew these plants looked alike, but were they really functioning the same way? And so we did experiments in Chile and California to demonstrate they actually were convergent in their physiology, their whole system physiology in terms of the allocation and so forth. And then we learned also that it depended on the sort of the light environment in a way. When we had closed canopy, the possibilities were less, whereas in Death Valley, we found there was such a diversity of adaptations going through the season, different plants coming at different times, but in the Mediterranean system, that wasn't the case. So that was an important step forward. But then I think it became obvious to me that the kinds of work that we were all doing in physiological ecology was crucial for the next level up, and that is, at that time, was a development in the early 70s of systems ecology, where they were trying to develop models for how whole ecosystems operated, and the information they needed came a lot from physiological ecology. And so I think that we joined up with the system modelers and helped we could do more as a physiological ecologist by integrating with the kinds of work they did. And then, as Chris says, and the next step was we could because of the fundamental stuff we were doing, it scaled so perfectly to ecosystems and then to the globe, and then we could work with the globe. And it meant that we could, through the process, we were working first with biochemists and physiologists and geneticists, and then we start working with the systems ecologists and then the geophysicist, and it's been a wonderful ride in the sense that what we were doing was relevant to all those different levels. It's just incredible.
But Hal, I think you're referring to the international biological programs and had different biomes and like grassland biome, probably that I was closest to. But that was an entirely different concept than the one we were working from. You know, we worked together because we liked together with a common interest, and we came from different fields, and then we tried to educate one another in different ways and helping out. Whereas the IBP programs mostly, not exclusively though, were very top down. You had an administrator on top, and you had a modeler, and the scientists became data collectors, which they didn't like to do. And I think that was a flaw. How is it now? We have a much better system now, I suppose.
Can I address that? Because that's an interesting issue.
Yeah.
The International Biological Program was carried out in the 60s and 70s. The main objective was to look at the productive capacity of ecosystems of the world. But there were two threads, and one was that I was involved in was the evolutionary. Why do these systems evolve to be the way they are, to look the way they are, to operate the way they are? That wasn't what the production people were doing. They were just looking at, what are the pools and what's the production. And so we had a very different thrust, and we did bottom-up science, and but yet the same principles of the plant as a package, their trade-offs and all that became very important in this evolutionary view of systems. So I don't think it was separate. Now, the IBP, the production people, they were all kinds of different schools. The one you were referring to was probably originally was the most successful. It was a top-down, and George Van Dyne was the director, and his idea was you could you get some modelers, then you get some people out there collecting the data, and then they sit in separate rooms and and there was stuff went up. And there was a rebellion against that midway through the program, and that didn't work so well. However, he had the best model, the best approach. I mean, they made great progress, and they broke that down, and I think it was still one of the more successful programs, although they never really achieved their main goal of understanding how that whole system works, and that came later. But I think the work going on in Europe and everything was more just collecting production data, pool sizes of nutrients, pool sizes of carbon, and so forth.
Yeah, I reviewed the boreal forest program, and it suffered from the same problems.
Right.
Top
down
yeah,
doesn't
yeah,
work.
right. Yeah.
Are we rerunning that with NSF and Neon?
Yep. You got it.
Okay.
One of to me, the high points, though, of the IVP was the meeting that both of you attended in Treban, in Czechoslovakia.
That was a crucial meeting. You want to talk about it? You got me invited to that meeting.
Yeah.
Thank you.
Well, the circumstances were not so good because this was in 1969, and it wasn't very far away from the tanks rolled into Prague from the Warsaw Pact, and the political situation was not very good, and the poor Russians we had there, common colleagues and so forth, were particularly uncomfortable during this conference. But scientifically, it was a real success. It was the time when we could get the physics, the chemistry, the biochemistry, the physiology, photosynthesis experts in different fields to actually talk to one another. I think it's the first meeting that these people will talk to one another in an informal manner and all of this. I remember especially Basil Cook, who was very famous at the time, of course, and he had done extremely advanced discoveries in the light reactions of photosynthesis. And he came up to me and said, "Do you believe in this C4 photosynthesis? Do you believe this oxygen effect stuff?" "Oh, yes." I said, "Well, you give me 5 minutes to convince me." "Okay, I give you 5 minutes to convince me." So I had to tell him about you can change the partial pressure of oxygen, and then this would change. And then he finally said, "Maybe you're right. Maybe the thing works." Okay. And then people started to talk to one another, and in a much informal and I think lots developed from that conference.
One thing that developed, I think, was crucial, was the was it Penning DeVries? No, who was it? No, who was the?
DeWitt.
Huh? DeWitt, yeah. That
DeWitt.
we brought the growth models there, and I think that was the first
Right.
time people really started paying attention to them,
Right.
and that had a big revolution on what we did, I think.
I remember he wrote on the blackboard, "No simulation without experimentation."
You're the genius.
The other thing that was, I think, a first at that conference was Otto Lange's work on showing that stomata responded to humidity, and doing that in the Negev desert, and with that really.
Remember, he was at that meeting. Was that?
The photos of his, I wasn't there, so I don't know, but the photos of his field system are
In that book?
In the book.
Okay. He was there then.
Oh, yeah.
A great colleague of ours.
Yes.
Yeah.
Well, one of Hal's other interests has been in invasive species, and that certainly is a point of departure for Australian science as well.
Well, there were invasions a long time ago, which have been rather interesting, and than the more recent ones. But yeah, it's interesting. You highlighted that Billings' entry into this was focused on the soil, and in Australia, there were 3 soil features that really strongly influenced the development of physiological ecology. I think the first one went straight into practical applications. That was mineral nutrient deficiencies. I guess the persistent one was salt and salinity. And when I was a starting graduate student, the major professor had the view that phosphate was intimately related to life forms, particularly the high phosphate requirements of rainforests and so forth. And he'd picked this up from a major up in Berkeley, Jack Major. And
Terrible equation.
well, I got exposed to it. It was just about my level of modeling, you know, conceptual but not quantitative. Right. Yes. But yeah, that and with that sort of background, it was assumed that plants of the same species, according to the taxonomists, that grew on different soils or in different habitats and looked different, were probably ecotypically differentiated, and there was never any genetics to back this up. But that's where I got into ecotypic differentiation into the salinity tolerance of two saltbush species, and it just happened that this was a remarkable system. The field work had been done by earlier students, and I guess I was concentrating on leaf anatomy, and of course that meant that I had to learn something about photosynthesis at some point, and it all came together. But when Chris was speaking about how long did it take to get the photosynthesis, C4 photosynthesis sorted, it took about 6 or 8 years for me, I think, to get things sorted. But yeah, these plants with unusual anatomy, which we didn't understand the relationship then to the pathways, just pointed one to a lot of very, very interesting studies. And again, people like Langer were at the time I was getting into independent research. He was hugely influential in terms of life form, environment, and physiology, all made possible by his contacts and his ability to convince Siemens that they should be providing the equipment for this discipline. And of course they did it for a few years and then said, hey, we can't make any money out of this. and fortunately he then managed to rescue that and see the firm Volz, which is with us today and is a major supplier, particularly of instrumentation for fluorescence measurements, which I guess is one of the quick and dirty ways to look at photosynthesis under natural environmental conditions. But soil, I guess soil was possibly the most easy to observe difference in many of these situations, particularly with altered andesite. I mean, there's nothing quite like that, is there?
Can I resonate on that a little bit?
Yeah.
That as I told you, we were looking at same climates around the world and looking at the vegetation and looking at the physiological response. Well, it all worked fine until you got to South Africa and Australia, because that's where soil characteristics, phosphorus limitation became overriding, and everything worked differently. What are all those ants doing here, for example? I mean, and it was very, very important. So we started doing studies among all the continents to show the role of soil. But then you bring up, I think, an important thing which hasn't been mentioned is the collaborative interaction between Lange's group, your group in Australia, which was, we exchanged scientists, exchanged ideas, exchanged graduate students, and it was a tremendously profitable, productive interaction. And unusual, I think. And
you had people like Ian Cowan,
I still haven't figured Ian out.
Well, he's still challenging. That's all. But and then a few bright folks like Graham Farquhar, who sort of came in and with a bit of guidance from Joe, got a few things sorted. Yeah.
Very important things.
Well, actually, you had not only the Canberra Stanford group, you also had the Canberra Würzburg mostly group, and also the Carnegie Würzburg group.
Actually, that's
so
an interesting triangulation.
Right. Mm-hmm.
Yeah.
And as far as the Würzburg group is concerned, it was primarily Ulrich Heber and Ulrich Schreiber.
Yes.
All of Otto Lange was involved in it.
You mentioned the starting collaboration with Olle in 1970 at the photosynthesis photorespiration meeting, but you published you hit the C4 photosynthesis rather early with a paper which you called beta carboxylation in Atroplex, which I guess it was published in 1967. But I noticed
69. It was 5 years after I'd done the experiments.
but that was a unique name. Olle in the 67 yearbook has a comparison of Atroplex species with and without beta carboxylation.
Well, I got that name from Harold Beavers.
Yes.
Did you? Okay.
Because he said, well, the beta carboxylation is well known from CAM.
CAM, yes.
And so I decided to use that term. And that's why I called the plant's beta carboxylation photosynthesis rather than C4 photosynthesis. But later on I went with the crowd. But
that's right. Yeah, it was the same in Australia. The one of the guy whose name I'm still not recalling, Joe, that suggested that it should be called beta carboxylation had worked in the same lab as Beavers in Newcastle on Tyne. Done his PhD up there.
Harry Beavers was really an unusual biochemist in the way that he interacted. He was very much a physiological ecologist before.
Very nice person.
Yeah, and had a great voice, and he used to sing silly songs about photosynthetic pathways.
That was perhaps the only conference I ever been to. It was the one in 1970 in Canberra, where we had symposium songs that were published.
Yes. No music, just the word. Joe, how did you get into all of this?
Yes, explain yourself.
Well, I was a chemist, I guess is my training. But I had the good fortune of being, at least through my high school years, living in Davis. And my summer jobs and sometimes during school was as a lab technician. And I did work on research projects that were being conducted by various people, including Bob Loomis. And he was unusual in the people that I interacted with, in that he didn't just want the numbers; he told me what he was doing.
Yes.
And he was figuring out how nitrogen nutrition influenced the sugar contents of sugar beets, and he could manipulate this by, of course, the nitrogen applications. And my role was to measure the nitrate levels in the petioles of these plants. So it got me very interested. He explained it so well that I figured even I could do that. I went on to study chemistry and got involved in things like steroid rearrangements and things that were quite a long ways from agriculture. And in my last year, I decided that I better get back, and I switched over to soil science for a master's degree. And in that, I encountered Hans Jenny and his influence. I didn't actually meet him, but I took the course that he designed. And I learned his soil forming equation and the ideas about how vegetation and soils interacted. And that was really formative for me. I thought that was just so amazing that you could tell what soils were present from a distance by what ecosystem was there. And in that process, I studied biochemistry and plant physiology, taking all the the prerequisite, and the graduate course that I was required to take. And it was an absolutely fascinating time. I was, after my master's degree, I looked around for a place to land, and was just extremely fortunate to be offered a position at the University of British Columbia with a fellow named of Bruce Tregunna, who was a co-discoverer, really, of the photorespiratory pathway, and also had discovered that corn was resistant to the oxygen inhibition. So he was very intimately involved in the early days of C3 photosynthesis inhibition by oxygen. And he and a graduate student who was working with me with Bruce Tregunna at the time when I joined him had been trying to put together what was going on with corn, and had realized that there was an anatomical association.
Right. Okay.
This graduate student was John Downton. Actually, both John Downton and Bruce Tregunna have passed away. Bruce died at the age of 36 with a massive heart attack after a touch football game. So he'd been really famous if he had lived on, I think. He was very brilliant. My role in this was to kind of, there was another graduate student looking at the interaction of CO2 and oxygen on photosynthesis. And I was sort of standing on the sidelines, and at one point said, hey, if you just increase the CO2 concentration, you wouldn't have photorespiration. Maybe C4 plants are doing that. And that was the hypothesis I started to use in my research. And fortunately, I met with Olle, who had discovered that what was necessary for this was for the RUBP carboxylase to be compartmented inside the bundle sheath away from the atmosphere. And Olle had actually discovered that this was the case. And that fit perfectly with the, I was trying all sorts of other ways to prove that. So he actually opened the whole thing up for me. And it was then a very straightforward thing to put together, how the CO2 was an intermediate in the C4 pathway. And very fortunately, I was also then able to get a postdoc to come to Carnegie to join Olle. And it's been a wonderful collaboration ever since.
It was, John.
It was, yeah. And I guess the other thing that was really quite important for me, I was a chemist, so putting things in equations was something I'd always learned to do. And when I got to Canberra, Graham Farquhar was an applied mathematics background, and I got together with people who were studying the enzyme Rubisco and its kinetics, and thought, geez, we could put that into some equations and make it work.
Yeah.
And it's turned out to be, it was kind of a fun thing to do, but it's turned out to be quite useful in the sense of
Very famous equation.
That's right. Very, very
It's being
central
cited very
equation.
often.
The funny thing was that it was really put together, the whole thing was put together to understand C4 photosynthesis.
Yeah.
We just made a C3 plant inside the C4 in order to we had to have something to simulate photorespiration.
Yeah.
And then we got to thinking, well, maybe it's useful all by itself. I think we've really covered the ground here. It's been a fascinating discussion, and it was a absolutely stimulating time in my life. The interactions with all of you, and I really appreciate the time that you guys have spent here and the interactions. Thank you very much for sitting with us.
Thank you, Joe.
Yeah, Joe pulled it all together. Yeah, thank you. Yeah.